You know, anybody who works on infectious disease, the ultimate goal is to get rid of whatever infectious disease you're working on and put yourself out of business. What we do in the lab is a lot of different research to support a huge international organization called the Global Program for the Elimination of Lymphatic Filariasis.
Steve has been working on lymphatic filariasis for a long time. He's a world expert on this, done amazing work. The idea that Steve's lab is pursuing is perhaps there are some natural products out there that people haven't looked at to treat filariasis that perhaps we can identify.
I work in a team of 6 people and we study 3 different plants, and we're trying to quantify them and their medicinal effects to see if we can potentially make a drug candidate to make a new treatment for lymphatic filariasis. So there's this big fear in the lymphatic filariasis community that the worms that cause lymphatic filariasis are going to become genetically resistant to the drugs that already exist.
When Steve approached me with this project, it was a great opportunity to move the collaboration beyond my lab into what's going on in Steve's lab with molecular biology. We talked about what his lab needed to do, what my lab would bring to it, and I think it's just an opportunity that makes a lot of sense.
The plant we study is Nerlina lobata, which is basically a roadside weed. What we're interested in doing with this plant is finding out quantitatively what compounds are in the plant naturally that are causing the worms to die.
So this is Nerlina lobata. We get it in huge shipments because Kristine needs a lot of it in order to make her extract, in order to isolate out the compounds that she spoke about.
What I'm really hoping to do eventually is figure out what are the bare bone basics of these molecules that are essential for killing these worms.
It's a really nice example of trying to find a solution to an important problem and then bringing 2 disciplines to bear on it. Us trying to help isolate the natural compound, and then Steve's lab to actually do the biological assays to see if it'll work.
This project sort of embodies part of the spirit of collaborations because we're taking molecular biologists and organic synthetic chemists and we're putting them together on a project that potentially could have widespread benefits.
I think Collaborations is a fabulous event, and it's something that we've been involved in since it started.
This is our data, and we found that
there's amazing collaboration going on across the college, and without a day like this, much of it would be hidden. And so I really like the fact that we bring it to the fore and celebrate all of the amazing independent work that students are doing in collaboration with faculty.
This drug we're looking at potentially kills the adult worms, which is what we really want. Because if you get rid of the microfilaria, that's great because you don't have as many worms anymore, but you still have the adults, and the adults can live in you for up to 15 years.
And there's no drug on the market that can kill adults in the human body. So that's why our plant is so great, actually, because potentially it's a way to treat lymphatic filariasis for people who already have the disease, because there's no treatment out there for people that are already infected.
So it could potentially wipe out the entire infection in your body. Yeah.
Chemistry has had such an amazing impact on life around the globe. And if we can play a role in that, I think that would be an amazing thing for me, for my students, and just to show how at a place like Smith with undergraduates, you can have a profound impact on human health around the world.